epithelial homeostasis, cell migration, and asymmetric cell division. And we're particularly interested in the power polarity network of proteins, and this is a conserved family of proteins across all animals, and they are crucial players in polarizing many cell types across metazoans. So we use the one-cell embryo of the worm as a model system to understand PAR-driven polarization. And so the PAR network, in this instance, is really composed of two different sets of proteins. And so and these two sets of proteins segregate into two opposing membrane-associated domains that establish the anterior-posterior axis this early on in embryogenesis. And so we can see in this example, in this embryo that is getting ready to go through the first zygotic division, where part 6 that belongs to one set of these proteins decorating the anterior side of the cell, and part 2 that belongs to the opposing set of proteins that occupies the posterior pole of the zygote. And so at the core of this polarization process is this principle of mutual antagonism between the anterior and the posterior pars. And so and what this means is that these two sets of proteins cross-inhibit each other from being able to bind to the plasma membrane. And so abrogation of this mutual antagonism, this PAR mutual antagonism, compromises the ability of the cell to divide, to polarize correctly. And so polarity, PAR polarity is an essential step at the beginning of worm embryogenesis, and we know it controls many downstream pathways. It controls the cytoplasmic polarization of phage determinants, and one such example is the phage determinant MEX5. It allows for the asymmetric division of the zygote, originating two cells at the two-cell stage that are different in size, where the AB cell on the anterior side is bigger than its P1 sister cell. And PAR polarity is openly very important for correct phage specification. And one way we actually use to judge a proper phage determination is by measuring the timing, the time difference between AB and P1 divisions, where the P1 blastomere always divides after the AB cell. And so, as I mentioned, at the core of this PAR polarization process, we have this principle of mutual antagonism between the anterior pars that antagonizing the posterior pars and vice versa. And so it is with this antagonism, this mutual exclusion between these two different pars implies is that the system has to be sensitive to perturbations in PAR protein dosage. Or in other words, the PAR system is sensitive to an imbalance of A-PAR-B-PAR ratio. However, we know that, and it's been established for decades now, that a lot of these PAR genes are actually haplosufficient. So a lot of these PAR heterozygous embryos can develop quite nicely into viable worms. And this is the example of, for example, of the gene PAR6. And we know that embryos that contain a single functional copy of this gene can develop quite nicely into a viable animal. And so this really prompted me to really get a good understanding of how gene dosage actually relates to protein expression, and secondly, how protein amounts actually translate and impinge on the ability of the cell to divide and polarize correctly. And so, of course, in order to do this, these correlations, we need to be able to measure gene dosage and protein amounts with high accuracy. And so, while it's perhaps a little easier to score a given genotype, measuring protein amounts has always proved quite challenging, especially when we want to accurately match protein amounts to a given phenotypic output. So we spent quite some time in the lab trying to develop this strategy that really allows us to quite accurately gauge PAR protein abundance in worm zygotes. So the first thing that we do is obviously perturb the levels of protein of a given PAR protein either genetically or via RNA interference, in embryos that express a GFP tagged PAR. We then use confocal microscopy to then quantify fluorescence levels in individual embryos. And this is aided by a strategy that we've spent quite some time on, where we can correct and discard this fluorescence signal from this inherent autofluorescence that we observe. I'm not going to have time to go into this, but if you want, I can tell you more about this later on. And so, thirdly, now we can that we can gauge better gauge protein amounts in individual embryos. We can now nicely pair this PAR dosage with a variety of phenotypic outputs on an embryo-to-embryo basis, as I mentioned. So if I now take you back to the heterozygosity that I've already mentioned, I told you that PAR6 heterozygous embryos are able to develop into a viable worm. And so this really prompted me to really get a careful analysis of how gene dosage actually relates to PAR protein amounts. And so in fact, at this instance, we actually sort of wondered whether in PAR6 heterozygotes, the expression and PAR protein amounts are actually dosage compensated, meaning that they're actually matching the levels that we see in the wild type cohorts. But this doesn't seem to be the case. And in fact, in our PAR6 heterozygotes, we see like a large drop in PAR6 protein amounts. And so, of course, at this point, we then started thinking about these results, and we really speculated that, worm development is actually resilient to a large variation in PAR protein amounts. And so really started thinking about this idea of system robustness that is likely inherent to the PAR polarization system. And so to really better address this question, I needed to really get a quantitative relationship between PAR protein amounts and a variety of polarity phenotypes. And really with this, really try to understand how animals and these worms can actually cope with such varying PAR protein amounts. And so focusing on PAR6, so one of these PAR proteins, the first thing I decided to do was to pair the amount of PAR6 dosage at the one cell stage with the ability of these embryos to divide asymmetrically. And so in this instance, the way I'm measuring this asymmetry is really by measuring this size ratio where between the two blastomeres at the two cell stage. And so a 0.5 ratio means that both daughters are going to be the same size, and higher ratios mean that the cell is able to divide asymmetrically. And so, of course, as you can already see here, these wild type embryos that I'm showing here in green are able to divide asymmetrically quite nicely. And so I can do the same analysis now for embryos that I treat with PAR6 RNAi for different amounts of time, and I can now populate the rest of the plot and really get a sense of how asymmetric cell division varies with varying PAR6 dosage. And just to give you an idea of what the two ends of the spectrum look like, on the top right of the plot, we see an embryo that is able to divide quite nicely in an asymmetric way. And on the bottom left of the plot, we see an embryo that almost lacks PAR6 expression completely, that divides completely symmetrically. And what I did after doing this dose-response analysis was really to sort of define this a range within which all these cells are actually dividing as asymmetrically and as nicely as the wild type cohorts. And so this really made us conclude that asymmetric cell division and the ability of these cells to divide asymmetrically is highly robust to a large drop in PAR6 concentration. Next, we decided to really measure and to determine how fate specification at the two cell stage depends on this PAR6 input. And as I mentioned before, we can basically measure this fate specification as a proxy, really, by measuring the time difference between the divisions of the two blastomeres. And as I mentioned, the P1 blastomere will always divide much after, a few 2.5 minutes after the AB cell divides. And so if I now perform a similar analysis to the one I'm showing here on the left, I once again obtain this dose response curve where I relate Parsec dosage to this mitotic asynchrony. In other words, to to correct to the ability of these blastomeres to define their fate. And once again, we see that for a large reduction, we see that these embryos are able to define, and these blastomeres are able to define their fate quite nicely, even with this large reduction in Parsec amounts. And so our work that is focused on this PAR-driven polarization in the worm zygote has really identified these nonlinearities in the relationship between protein expression and a variety of phenotypic outputs. And more specifically, I've shown you that asymmetric cell division and fate determination of the two blastomeres that result from the first zygotic division are highly robust to large perturbations in PAR dosage. And so the ongoing work in the lab is really to try and understand how this robustness property is implemented or embedded in the PAR polarization system. And so there's various possibilities that we're exploring at the moment, but basically it's possible that there's various regulatory feedback mechanisms that allow for system control, and or there are redundant pathways that actually cooperate along with the PAR network to ensure phenotypic robustness. So I'd like to thank you for your attention, and everyone in the lab, and everyone at the CRIC, and I'll be happy to take questions now or later. Thank you.
Great, thank you. Thank you. That was great. So we have some questions from the Q&A. First question is, why was Parsec tested first? Was there an a priori reason to think it might be the component that is dose dependent?
No, so we've actually performed the same analyses for a lot of the other genes, and in fact the picture is quite similar for a lot of the PAR genes, irrespective of being on the anterior or the posterior set. But a lot of them are showing this. Basically, I do identify this robust property for a lot of these proteins actually.
Great, and then we have someone asking about your autofluorescence correction. Can you share how it works?
Well, yeah, it's a little involved, but I can definitely, if I get their contact, I'm very happy to share that. It's a very simple method, but it would involve perhaps another 10 minutes. But yes, I'll be happy to share. Yeah.
Okay, great. And then finally, last question we have time for is: does PAR dosage affect PAR polarity, i.e., the size of the PAR crescent? Did you actually measure how much is getting loaded onto the cortex?
Yeah, so I actually, I do have that. In fact, perhaps what you can see here is that when I actually measure the domain size, so that really the ability of these cells to polarize, I see that once again, for a wide range of Parsec dosage, these embryos can actually form a similar size crescent, just like the wild type embryos. So it is possible that it is really down to this ability to concentrate protein on the cortex that then ensures correct phenotypic outputs.
Great, thank you so much. That was really great.
Thank you.
Let's move on to our next speaker. So we have Joshua Abrams from the NYU School of Medicine. He'll be talking about the role of polarity proteins in intracellular tube expansion through April recruitment of the exoskeleton complex. Sorry. So Josh, if you can share your slides, thank you.
That looks good. Okay.
So we don't see your slides.
Oh. Let's see.
I don't see your slides.